Welcome everyone to the seminar series, Plant Breeding. It's my pleasure to introduce the first speaker, Matt Carroll. Matt is a PhD student with my group. He started in his PhD program in 2017. He graduated with a bachelor's of science in genetics as well as in agronomy with the plant breeding option in 2017 from Iowa State University. He's going to be talking about one of the thesis chapter that he's been working on, but he's also worked on a number of other projects, primarily that relate to drone-based phenotyping. He's done a short internship in Japan, University of Tokyo, and it's my pleasure to welcome Matt.
Thank you, Danny, and thank you all for attending the seminar today. Today we're going to be talking about a paper that we've recently submitted to Plant Phenomics, Deep Multi-View Image Fusion for Soybean Yield Estimation. So a little bit about this project and its background. In 2015 and 2016, we had a couple of cohorts of mechanical engineering undergraduate students who, for their capstone projects, worked on a ground-based phenotyping buggy for our lab. This initial work eventually led to an NSF USDA-funded grant on ground robotics and phenotyping. And this work was done in collaboration with Dr. Bhattacharya in mechanical engineering and his PhD student, Hyung-Ching Gao. On the right here, you can see just an example of that ground-based robot and traversing the field. As you can see, it does pretty well in muddy conditions, and that's kind of one of the benefits of it. This is one of many ground-based phenotyping efforts that we have done in our lab with the Soynomics group. And so what I'm presenting today is in collaboration with Danny and myself and Dr. Sarkar and his PhD student, Luis Riera, as well as the other Soynomics PIs. So some previous work and motivation and why we're interested in using pod counting for yield estimation. Any of you who have worked in plant breeding know that harvesting yield trial plots is a very time-intensive and labor-intensive task. And not to mention that weather can also be a very limiting factor of getting in the field. This picture was taken a couple days after that previous video that you saw. And we had been out of the field for about 2 weeks, and we thought that maybe we could go, but it was just a little too muddy yet to get into the field. Previous work in our lab has focused on in-season yield prediction. Jonathan Shook, who's now at Corteva, he worked on using pedigree information as well as weather information to predict yield. And then Kyle Parmley, who's now at Bayer, has worked on canopy reflection and some other above canopy traits for hyperspectral yield prediction using indices such as NDVI. One of the reasons that we want to do this in-season yield prediction and end-of-season yield prediction with the pod counting is that we have this dream in our group that we only harvest seed plots for advancement. And so we could do this at a single location. In our case, it'd be the central location here in Ames. And then all of our outside locations, we need some sort of form of estimating yield accurately. And this would greatly increase our ability to both harvest more plots and basically analyze larger populations or more crosses per year, which helps increase our genetic diversity that we're scanning in a given year. Plant organ detection is a difficult trait to collect, but other crops have shown progress in it and good results. These have primarily been in horticultural crops such as grapes and oranges, where they have larger plant organs as well as a higher contrast between the foreground and background. And what I mean by that higher contrast is that in the case of oranges, the color is very different from the background, so you're able to easily segment that with a color thresholding mechanism, then estimate the number of plant organs that way. With soybeans, of those of you who are familiar with the physiology, the pods tend to be the same color as the stems, so using a color thresholding mechanism doesn't really work that well, which creates another obstacle to overcome for this sort of computer vision task. Here, we're going to be talking about the data sets we're presenting. We have two data sets that we work with, the controlled imaging data set and the rover-acquired imaging. The controlled imaging was captured kind of as our preliminary trial for this, just to see if we could even do anything with pod counting and detection. And the rover-acquired data set was more or less moving into that real-world type of scenario that you're going to be needing for any sort of scaling to a soybean breeding program. What you're seeing here is just the training and testing set distributions in both sets. They both follow very similar distributions, as is important with any sort of modeling that you're doing. If your training set doesn't and your testing sets have very different distributions, you're not going to get the results that you're looking for. Now we'll get into the image and data acquisition. On the top row here, you'll see the controlled imaging situation. And what you see here is that we have this black trifold background, and this helped to create a barrier to basically make it easier to segment out the plant from the background. In addition to this, we took images from the left, center, and right of each plant, and we took these with a very high-powered camera, the DSLR camera, so we had very good resolution. In addition to this, with the controlled background, we also tried to control the lighting to the best of our ability. This was still captured outside, so there is some variation, but we take out an umbrella with us to help kind of block out the sun. You can see in this case, we didn't do the best job, but for the most part, we had a very good high-quality image data set with very little noise, or as little noise as you can have with it still being an outdoor imaging environment. This was actually collected in 2015 when I was an undergraduate research worker for Danny. And so after we would collect this data and image it, we would then hand harvest all of the plants within the plot and very carefully and meticulously put these all into a bag, a mesh bag, so that none of the pods would fall out or the seed. And then later that fall and into the winter, we went back and individually counted every single pod for each plot. And then Danny asked me if I wanted to still do my PhD here. And for some reason, I said yes. I'm joking, of course. This was a really great experience to learn how to conduct research and the importance of collecting high quality data. Then we'll move into the rover acquired imaging data set. This was taken in 2018 in Jonathan Shook's field. These are hill plots. And what you see here is that these were collected using a Logitech webcam to collect videos. These did have some challenges in the sense that the image resolution was lower quality. This was made up for marginally though, because the imaging was much closer. The pod and the pixel sizes were actually very close to each other for both data sets. But what you do see here is that when you're that close, you do sometimes have some cutoff on the top, and so you're not always capturing the full plant. But the good thing about soybeans is that most of your yield isn't on the very top of the plant. Most of it's in that lower 2/3 of the image, or as you can see here. In addition to this, in this situation, we did have pretty consistent lighting conditions, but with an outdoor imaging data set, when you're going up and down different sides of the rows, you're going to have changing weather and lighting conditions, which can add another layer of complexity and noise to your phenotyping system. And here is just kind of an example of the phenotyping robot. These green blobs here are basically the hill plots, and what you see is that we had cameras on both sides, and this just helped to increase the throughput of our image phenotyping system. And as you can imagine, we gave up some image quality with this phenotyping system, but the benefit is that we could, we could phenotype many more plots per unit time than we can with that controlled imaging setup. Then we'll talk about data augmentation. This is really only applicable for the image required data set, or the rover acquired data set. The reason for that is that for the controlled imaging data set, it was clean enough that we didn't have that much noise where we needed to do any sort of data augmentation. It performed quite well on the original data set. And in ML or any computer vision problem, a common way of doing data augmentation when you have images is to do some sort of image histogram stretching, or maybe doing some sort of rotation. We did try that, and it didn't work out as well as we had hoped for. But then we realized that we're capturing all of this data via videos at 30 frames per second. And what that means is that every second you're rolling this video, you're capturing basically 30 individual pictures. And so with those 30 individual pictures, we thought, okay, so we can just more or less, we got multiple views of the plot at multiple time frames. So what we did is we took our 123 plots that we had, and we divided into 78 of our training set and 45 for our testing. And then we went through and randomly shuffled the frames that we had on those 78 training plots, and gave us kind of an artificially inflated testing size of 234. We found that this worked really well for us. The reason we did 3, so we did try larger iterations, but we find that if you go too far with this, you actually end up overfitting your model, and it performs worse. So for us, the optimum was at that 3 permutations of that image shuffling. For different computer vision problems, it may be different than this. And then we'll get into pod counting as a yield predictor and why we're using that as a proxy for yield in this situation. You'll see here on the x-axis we have pod counts, and on the y-axis we have seed weight in grams. And you'll see here that in the controlled environment, the R-squared is 0.82, and in the rover acquired data set, we have an R-squared of 0.76, showing us that in both situations we have very good, high correlations with pod counting and seed yield. I should point out that in the rover acquired data set, we did the soy NAM parents, and those of you who are familiar with the NAM parents know that they have both PI lines, diverse lines, and elites. And so we had a little bit more diversity in our training set there, but we did that intentionally because we didn't want it to be, we want our model to be robust. We didn't want it to be only capable of looking at higher yielding lines or elite lines. We wanted to be able to scan the entire genome. On the controlled environment side, because this was our initial testing and we weren't entirely sure how well this would work out, we used the checks within our breeding program, which all tend to be, public elite lines. So a little bit more consistency on, the number of seeds per pod, seed size, and seed weight, things like that. And that's why we think we're seeing a little bit of a discrepancy in the R-squared values for pod counts to seed weight in these two data sets. Now we'll get into the multi-view image fusion model and how that works. This is actually a two-step model. It focuses the first step being on detecting the number of pods in each image that you feed into the model. Excuse me. And then the true pod count estimation is the second step, more or less regressing the values that you had from the first step to a final pod count. What you see here in the video is a plant tracking algorithm. So the large red boxes are identifying plants and tracking them throughout multiple video frames. And then the small red boxes are identifying individual pods on each plant and giving a probability estimate of how likely, the identification that it's saying it actually is. These two models were not integrated in this paper, but for future work, it's something that is going to be necessary. The reason for this is that as we move into yield trial plots, there will be many more plants per unit area. And so the overlap between plants is going to be quite high. So having that plant tracking methodology so that you're not double counting plants is going to be essential. In addition to this, it's also going to help with the optimal frame selection. Right now, we went through and manually labeled the start and end of each plot and then kind of randomly selected frames based on the time series. That's not going to work as well when you have those plants that are close together. So having a plant tracking algorithm will allow us to basically select frames that don't have additional repetitive data so that we can get the best estimation model without double counting pods for future work. Now we're getting into the first part of the model, the training and pod detection model. What you see here in green are the human rater annotations. What that means is that myself and other members of our team went through and manually drew boxes around every single pod that they saw in an image. At this point, I'd like to thank Clayton, Sarah, Lisa, and all the other members of our team who took the time to label. It's a very time-intensive and very tedious task, but they did an amazing job with it, and we got good results because of the quality of the labeling. The orange boxes are the output from that model, the pods that were detected. And I think it's worth pointing out that in this plot here, the labeler only labeled the foreground, the pods that we were interested in. And then there was also this background noise that very easily could have also had pods labeled, but there wasn't. And that's just because of the quality of the labeling that was done. With a lot of the supervised learning, if you don't have quality annotations, your output's going to be quite poor as well. So the quality of the annotations was very important for this, and I think we got that. Now going to the VGG19 model and how it works and what it does. It's based off of a convolutional neural network, if you're familiar with those, or a CNN. And what a lot of image classifiers do is they're very good at classifying one class per image. And so an example of that would be if you had a data set of images that had, only corn or only soybeans within those images, it'd be very good at saying, this image has corn, this image has soybeans. The VGG19 model is a multi-class detector. And the difference in that is that it can detect, multiple classes within a single image. And in addition to that, it also maintains the spatial awareness of where those classes are. And so in our situation, we only have a single class, which is pods. But we have a very vested interest in knowing where were those pods at and how many were there. And that's why it works very well. And so walking through this model, you'll have a single image that's in the input for this phase one. It runs through a convolutional neural network, and essentially these red boxes are representing the high-level feature classes that show the importance of where it's detecting pods. And we maintain these throughout for the next step in the model. And the eventual output of this is these orange boxes, that output of how many pods were there and where were they at within each individual image. The next step is the multi-image data fusion. And so this takes the features extracted from the pod counting data and uses a CNN with 3 layers to predict the pod count of the plot. So what you see here is, so in this particular plot, we had 6 total images of that plot. So each of these individual images ran through that phase one of the model, which is represented by this box here. And then once those are all done, this part of the model then takes all of that data and runs it through these convolutional neural networks. And these feature classes are represented by these multi-layer arrays that have pretty high dimensional data. And running through these convolutional neural networks, we then get those multi-dimensional arrays condensed down into a single layer vector. That vector is then run through these dense, fully connected layers, which eventually regresses those values to the estimated pod count. And this is a good way of kind of showing that the difference between what we've done with this plant organ detection compared to other products or other approaches. These estimated pod counts are not, what the raters saw in each image and labeled it as. It's the pod counts that we manually took on every single plot that we harvested. So this model is, taking what it can see, but then it's also trying to estimate the occlusion parameter of saying what can't we see, because we know that no matter how many images we take of a plot, there's still going to be pods that we can't see, and there's going to be that yield component that we're missing. And so we're trying to train this model to more or less estimate what it can't see based on what it can see and get an estimate for the final yield. With that, moving into the results, you'll see here the results for the control set. In the lower row is the rover acquired image data set. I will clarify here that the one image for the control set is just a single central image. Whereas the rover data set, one image means one image per side, so it had a total of 2. With that same logic, the control set with 3 images had 3 total images, whereas the rover acquired data set had 6 total images with 3 per side. What you do see with the one image is that it does increase, the accuracy increases from 0.698 to 0.711 in that control set, and the RMSE reduces, which is what we are hoping to see, and what we would expect is that more data, gives you better results. Not surprisingly, the control set did perform better than the rover data set. That'd be expected with the level of noise and the kind of the data set sizes. One thing we're hoping to improve with future work is you'll see here on the x-axis is that we have a range of about 150 to 1,100 pods, whereas the predicted values range from maybe 300 up to 650. This is once again partially corresponding with the smaller data set size. We think with a larger, more robust data set, we could, get this line to be more of that one-to-one matching that we're hoping to see. But as a breeder, my primary focus isn't always on exactly how well I can estimate yield. It's more on how do I rank my lines, and can I accurately select the lines that should be advanced and accurately discard the lines that are not my top performers. And so here we're representing just the rover acquired data set, so the uncontrolled data set, using 20 and 30% selection thresholds as kind of the estimates of what a breeder might use for their selection threshold, saying I only want to keep my top 20% or top 30% of lines. And what I have here, the equations for accuracy, sensitivity, and specificity below, they're just kind of a reminder of what these metrics mean. They're something that we all use and see quite a bit, but if you haven't looked at them recently, sometimes it's good to have a little reminder. What I would like to point out is that with the 20% selection threshold, we didn't see much improvement in the metrics, but when you move to a 30% selection threshold using 1 versus 3, we saw improvement in all metrics. And so showing that here, with the accuracy starting out, we had 0.69 for one image and then moving to 0.78 for the 3 images. With sensitivity, we see a movement from 0.46 up to 0.62, and with specificity from 0.78 to 0.84. Couple of things to point out here. Your sensitivity is kind of your true positive rate. So thinking about it, if I make a selection, say that a line should be advanced, that was actually the correct decision, and that line should have actually been advanced. And then looking at the specificity rate, that's more of looking at your true discard rate. So when you say that a line should not be advanced, it actually should not have been advanced. And these are all based off of once again that ground truth data of the manual pod counts and the ranking with those. To point out 2 things with this. All models seem like they performed fairly well on the specificity side of things. So they all seem to work pretty good on the discards. Something that we're working to improve is that sensitivity metric. But on top of this, these were this is a relatively small testing set. And so when you're running these smaller testing sets, a few changes in the classification can really change your values. And so in future work, we get, larger data sets will have a little bit more of a robust, more accurate estimates of these true values. So talking about conclusions, the 2 main things from this paper were that using ML, we can overcome the occlusion problem as well as the noise of outdoor data. And what we mean by that is that, we can accurately, fairly accurately rank results based off of, what we see and what we can't see, which is a big problem both on the agronomy side, but also on the machine learning side of things. How can you, estimate what you can't see? And then on top of this, it's a different way to estimate yield within yield estimation of remotely sensed data, most people are, doing some sort of vegetation index, like Kyle has done, and those all work very well. But this is something different, and we think maybe has some potential for, late season yield estimation instead of early season yield estimation. And then moving forward in future work, we've got a few things that we'd like to work on. Part of it is just moving to more off-the-shelf rover phenotyping platforms. This will allow us to maybe have more consistent imaging as well as higher quality images. On top of that, we'd like to move to onboard yield estimation. This is important because we need quick turnaround time and decision making as a breeding program. And so right now, when we collect this data, we bring it back to the lab and then do all the analysis there. That's not going to work as well when you're trying to make quick decisions for advancement and selections, and that's going to be done by integrating that plant tracking mechanism on these rovers in the future. We'd also like to move to try doing this UAV acquired data. We collected some data in 2019 for that, but we didn't get it in 2020 because COVID has just been a fun year for everyone. But in 2021 we'll do that. One of the issues with UAV acquired data is that the resolution is going to be very different from an aerial-based phenotyping mechanism from the ground-based. But the good thing for us is that cameras keep getting lighter in weight and higher resolution images. So we're thinking that we eventually will be able to overcome that problem. And the reason we'd like to move to UAV acquired data is the scalability of it. Right now, rovers are capable of covering maybe 5 acres in a given day, whereas a UAV, it's pretty reasonable to estimate that you could cover about 5 acres in an hour. So that's just the scaling capability of it. And one last thing is moving away from pod counting as a ground truth for combine for our ground truth. The reason for that is, once again, counting pods on a hill plot that has maybe 5 plants total, we can do decently well. But when we start moving to yield trials that have 100 plants or more per plot, that's going to be really difficult to maintain that scale, especially when some of our initial tests have 10,000 plots or more. And so with that, I'd like to say thank you to all of our funding sources who, without them, we wouldn't be able to do any of this work. And then also to all of our lab members who have helped with labeling, data collection, and everything else. So thank you. And if you have any questions, I'd be happy to try and answer them.
Okay, if anyone has any questions, just unmute yourself and ask, or you can ask in the chat.
Hi, I have a question. Can you hear me?
Yep.
Oh, this really excellent research and nice results. So one question is, maybe I start with my background. I was a commercial soybean breeder over 10 years right now as an alfalfa breeder. So the first question is, you asked me the grain yield. I wonder how well the accuracy for the biomass yield, since the rover can measure the whole plants, and how will this can be used for alfalfa biomass? Number one question. Number two question is, the technology is really good. As a soybean breeder, we needed to connect. We need to harvest the soybean. We will get a yield at the same time with a new combine or any tractor. You can get a yield quickly. Which stage do you think this technology, this research will be used for to increase the genetic gain or speed up your breeding program?
Yeah, so there's some good questions. I haven't really thought about this for use in non-soybean crops, but as far as estimating biomass, I would say that you could take this approach and then combine it with a technique called structure from motion, which is a way of creating 3D reconstructions of objects using 2D images. And so I think from a biomass estimation standpoint, I think you could maybe do it better with a drone. But with this, you could also probably run this through plots and kind of get a 3D volume of that for a biomass estimation. And then to answer your second question on where would we see this being used within a soybean breeding program, and you're right. So we do get yield back very quickly on combines. The reason, but where we see this being used at is more so that you can kind of condense where your combines are at. So you have a central location where you can advance your, you harvest every single line, and then you get your yield estimates, but you also have your seed for the advancement. But then at your outside locations, if you can get accurate yield estimation using this, you don't have all the logistics of hauling combines to different locations, as well as harvesting every single plot. Every plot for us at least takes, you estimate about 20 to 30 seconds per plot. And so we can go through that a lot faster with these, rovers or maybe UAV acquired data, which would allow us to get through those locations, I think, in a quicker fashion than we could by just combining them for every plot for yield.
Yeah, thank you. So the one you mentioned for the biomass yield using UAV from the air, we use this rollover from on the ground. How that resolution and how when you fly the UAV, you need to control the head for the resolution. Can you comment using the two as to the resolution?
Yeah, so the resolution on a drone, and it's going to be very dependent on your altitude, but for us, if we fly at 60 meters, so 200 feet, our our resolution that we're seeing is half a centimeter per pixel. On ground-based phenotyping, I don't have the exact measurements, but it's getting into the couple of millimeters. Like it's less than that. It's getting into the millimeters per pixel, not centimeters per pixel.
Okay, great. Thank you.
Hello, Matt. You have an image to showing that the labeling by the grad students versus the computer. But when you're doing ground truth, did you actually harvest those pods to count exactly, or how do you actually have the ground truth instead of image?
Yeah. So for the training set. So we actually imaged for this rover acquired data set, it was probably about 3,000 total plots. And so for the image labeling portion, not every single plot had a yield associated with it, but for every plot that we actually then did the analysis on for both the training and testing, those all had where we manually harvested each plot and then went through and counted every single pod within that plot. It took a lot of time.
Work. So, okay, another question that when you actually, let's say, if you counted some plants, and then do you think that counting procedure eventually will help the labeling procedure? That if people actually counted, they actually get to learn how to label more accurately? Would that be helpful? It's not a critical component.
So asking about how large of a labeling data set you need for the pod annotation portion?
No, if you actually experience the counting process, exactly, the actual counting process, does that feed back to your labeling process?
That's a good question. For the labeling side of things, grad students and myself did all of it. So we all had kind of, we help each other with our research. So I think everybody did a very high quality job with it. On the accounting side, when I'd go back and check some of our undergraduate workers' work, usually the pod counts were within I had ranges of 2 to 5. So I was very happy with the level of accuracy that students were giving me for the results.
Okay.
But I think if you've counted, you're very you're gonna have you put in a lot of blood, sweat, and tears at that point of counting pods. So you want the data to be good.
And Jenring, that's a good point. Just to add, as Matt said, they were sort of double blind, so to speak. Matt went back and made sure that all the undergrad students and all counters, that their work was up to the quality that was needed. So they knew that double check will happen.
Yeah, I'm mainly asking if you actually counted and then you see the image, then you might labeling more on the back, on the not the just front, on the back.
Oh yeah, no, those so those were separate. It's like when you were labeling, you had no idea how many pods were estimated for that plot. So there shouldn't be any bias associated with that I'm aware of.
Okay, good.
Hey Matt, great presentation. How many images did you say again were used to train the model?
So it depended on the model itself. So for the pod annotation portion, I think we ended up having 523 images that were labeled for that portion. For the yield estimation portion, it ended up being the 200 and let me see if I can go back to it. I think it was 240, yeah, 234, but it was actually 78 plots. But then we did that permutation of how many iterations we ran through for that testing size. So it's a smaller data set, but still worked quite well for the results that we were having.
Yeah, definitely. And yeah, that was more than I thought. I was thinking you only used 234 for the pod part. And this model was trained. It was a new model, right? You didn't build it upon an already trained model and just recalibrate it.
No. So the VGG19 was actually, it's pre-trained on RetinaNet.
Okay.
But then we take that pre-training though, but then we go through with our own labeled pods and do a final training on it with it. So it's kind of pre-trained on that RetinaNet model, but then for the actual feature extraction, it uses the labeled input that we had for it.
Okay. Thank you.